this session is an interesting one because we're going to highlight the subjects of genomic and epigenomic studies. I have the honor to present a dear colleague and one of the early founders of the Center for Arab Genomic Studies and an eminent academician at Al-Ain University, Assistant Prof. Dr. Ahmed Al-Marzouqi from the Faculty of Medical and Health Sciences in the UAE University. So he is also Assistant Dean for Scientific Research and Graduate Studies at the university. He got his PhD in Biochemistry and Molecular Biology from Pennsylvania State University. Of course, he looks at things from the detailed point of view, so looking at the subcellular organelles. I always admired his lectures, the way he gives such complex subjects in a very easy way. His research focuses on chromatin modifying complexes, nucleosomal modeling, published many papers in peer-reviewed journals, as well as received awards and honors. Let's welcome all Dr. Ahmed Al-Marzouqi to give his talk on the molecular functions of chromatin modifiers.
Thank you, Mr. Chairman. Good afternoon. How are you doing? Well, my talk is going to be a little bit different. Today, I'm going to be talking about epigenetics and in particular, the molecular functions of chromatin modifiers. So please feel free to take a nap, especially after that great lunch, okay? Okay, let's see. Okay. I'm going to just, in the next probably 25 minutes or so, I'm going to talk about what my lab is interested in doing. This is the building where my lab is based in Al-Ain, next to Tawam Hospital. This is the College of Medicine at the UAE University. Okay. As a lot of you already know, eukaryotic DNA is packaged to form what are called nucleosomes, wrap around an octamer of histones to form these nucleosome molecules, which in turn wrap around themselves to form these higher order structures and eventually the metaphase chromosome. This higher order structure of chromatin is a repressive structure to many biological processes such as replication, transcription, and recombination. So if you look at the nucleosomes under an electron microscope, you see what look like beads on a string. So this structure is called beads on a string structure. As I said, this structure is a repressive structure. So many transcription factors that are required to turn a gene on can no longer bind when histones are bound to DNA, okay? So this structure is a repressive structure. So transcription factor TF2D, for example, cannot recognize the TATA box, and hence RNA polymerase is no longer recruited to activate this particular gene, for example. It's as if you want to drive to Al-Ain from Dubai with your car and you come across a group of camels on the road. Obviously, this group of camels will need to be moved out of the way before you can progress. So RNA polymerase needs the histones out of the way before transcription can be activated. Fortunately, in the cells, there are groups of proteins that do this job. These are called chromatin modifiers. So the job of these chromatin modifiers is to relieve chromatin-mediated repression. This repressive effect of chromatin is relieved as a function of these chromatin modifiers. These are large molecules from hundreds of kilodaltons in size to megadaltons in size that are recruited by transcription activators to exert their effects and eventually lead to transcription activation or silencing. Now, there are two classes of chromatin modifiers. One is a structural modifier that use the energy of ATP hydrolysis to exert their effects to remove the histones out of the way. And secondly are covalent modifiers that covalently modify the histone tails, and by doing so will lead to transcription activation or repression. So we're going to be talking about histone acetylation. This is a mark, an epigenetic mark on the histone tails that lead to transcription activation. It's a reversible effect. These acetylation marks can be removed by histone deacetylases to repress certain genes. And obviously there are also phosphorylation and methylation of the histone tails as well. So let's talk a little bit about the first group. Those are the structural modifiers that use the energy of ATP hydrolysis. They bind to DNA and nucleosome, and by doing so, they break histone DNA contacts using the energy of ATP hydrolysis. So these are huge molecules, and this is a nucleosome that wrap around the nucleosome and try to affect them, remove them, for example. They work in a variety of fashions. They either move the histones in cis along the DNA or completely transfer the octamer to another piece of DNA, hence releasing the DNA, and transcription factors can now bind. They can also basically exchange a dimer. So they could exchange H2A, H2B dimer from the histones, for example. The covalent modifiers, as I said, they covalently modify the histone tails. So if this is the nucleosome, the histone tails are protruding outside the structure of the nucleosome, and these tails can be modified by acetylation, methylation, phosphorylation, or ubiquitination. All of these are post-translational modification signals on the histone tails that have specific functions. So what happens is that when these tails are, for example, acetylated on histone H3, certain groups of proteins can come and recognize these flags and additionally exert their effects to turn a gene on, for example. Or for example, when these histone H3 tails or H4 tails are methylated, a different class of proteins can come and bind and again exert their effects, either activation or repression. So histone acetyltransferases are recruited by activators that are bound at the upstream activation sequences to turn on a gene, and histone deacetylases can be recruited by repressors to turn those genes off. There are also other chromatin modifying proteins such as methyltransferases that are recruited to have certain effects as well. This slide shows a cryo-EM pictures of some of these chromatin modifiers. This is the crystal structure of the nucleosome. This is RNA polymerase II, and as you can see, a lot of these chromatin modifiers are huge. They are much bigger than a nucleosome or even bigger than RNA polymerase. So this is an example of a histone acetyltransferase. It's called SAGA, and these two are examples of chromatin modifiers that use the energy of ATP hydrolysis to remodel chromatin. There's a lot of evidence of the link between these modifications and the epigenetic signals and cancer. So for example, DNA methylation at the promoters have been linked to cancer. So there is a lot of DNA methylase inhibitors that are used perhaps in therapy or at least in clinical trials. HDAC inhibitors are also in clinical trials. So HDACs are overexpressed in many cancers. Many subunits of chromatin remodelers are also mutated in a variety of cancers. Histone acetyltransferases are mutated in some cancers, as well as phosphorylases, kinases, and methyltransferases. There's a link for some of those to cancer as well. This table shows a variety of HDAC inhibitors that are currently in clinical trials, or DNA methylation inhibitors that are in clinical trials. So what I'm going to do in the next few minutes, probably 10-15 minutes, is talk about what we are interested in my lab. So previously, we had shown that different chromatin modifiers can function together to have a synergistic effect. In this paper, we kind of showed that acetylation by either a histone H3 acetyltransferase SAGA or an H4 acetyltransferase NUA4 can affect the retention of another chromatin remodeler called switch SNF to templates, to promoters. So in the presence of acetylation, this complex is recruited, but not in its absence. The same result is observed when an H4 histone acetyltransferase is used. Again, an ATP-dependent chromatin remodeler is recruited in the presence of acetylation, but not in its absence. Later on, we showed that a specific domain called bromodomain within this ATP-dependent chromatin remodeler is important for this retention. So when this bromodomain is deleted, the retention is completely lost. More recently, we have shown that this same bromodomain is required for the displacement of a histone acetyltransferase SAGA bound to acetylated nucleosomes. So the idea is this, that histone acetyltransferases are recruited by activators to promoters. They acetylate the promoters, and then that will lead to the recruitment of other chromatin modifiers, such as ATP-dependent chromatin modifiers. So it's a two-step reaction leading to activated gene expression. We use, for all of our assays, we need to purify these protein complexes, native form of these protein complexes, and the way we do that is through a tandem affinity purification technique where the protein of interest is tagged. It has a protein A tag that binds to IgG beads. It has a protease cleavage site which then can be cleaved, and it also has a calmodulin binding peptide that binds to calmodulin beads which later can be eluted. So it's through a two-step affinity purification step that we purify most of our protein complexes. Here's a silver stain of a switch SNF complex and a histone acetyltransferase complex. And as you can see, multiple bands on a silver stain. This shows all the subunits that are part of these complexes. Here's the Western. So our goal in the lab has been to basically identify novel chromatin remodelers and study their functions in vitro and in vivo. One of the proteins that we have recently been working on is called FUN30. It stands for Function Unknown Now 30. Okay, it's a protein that was identified based on sequence homology to the ATPase domain of known chromatin remodelers. The mouse version of this protein is called ETL1, and in humans, this protein is called SMARCAD1. It's a non-essential protein, meaning that if you delete the gene, the cells survive. In mouse, it is widely expressed, and deletions can cause a significant developmental phenotypes. Okay. We've published the results of our in vitro work not too long ago, so I will not get into that. And more recently, a couple of papers in Nature talked about the role of this protein in double-stranded DNA repair and DNA end resection that is required for double-stranded repair. There was also a paper more recently about the role of FUN30 at centromeres. So it seems that FUN30 is also targeted or localized to centromeres. And there is also some evidence that it might have a role at origins of replication. So we were interested in identifying the role of FUN30 at origins of replication. This is a growth assay showing that the deletion of FUN30, in the deletion of FUN30, the cells become resistant to UV irradiation as well as to ionizing irradiation. So whereas wild-type cells die fairly quickly, the deletion is become more resistant. The same is apparently true if we use a topocyte. The deletion of FUN30 also arrests cells at G1S phase. So this is the wild type strain, and this is a deletion where you see a delay in the transition from basically G1 to S. So also, the deletion causes an increase in the expression of a lot of proteins that are activated during cell cycle arrest. Those are RAD53, RAD9, as well as CDC28. So the deletion leads to an expression or an increased expression of phosphorylated version of RAD53, for example. If FUN30 has a role at the origins of replications, it could possibly have some activity such as topoisomerases, and therefore we tried to see whether FUN30 can relax DNA that are supercoiled. So here's the DNA that is negatively supercoiled, and this is our positive control. What you can see is that with increasing concentration of FUN30, this band decreases and that band increases, meaning that basically negatively supercoiled DNA has been relaxed by FUN30. This does not seem to be ATP dependent, as they, or in fact may be inhibited by ATP to some extent. We wanted to also see whether what is the function of FUN30 on positively supercoiled DNA. So what we did is we started with the we wanted to see whether we can generate positively supercoiled DNA. So we started with relaxed DNA, and in the presence of FUN30, we were able to generate positively supercoiled DNA. So it seems that FUN30 can take negatively supercoiled DNA, relax it, and even go further to generate positive supercoils. It's a function that might be required at the origin of replication. We also tested, this is another in vitro assay showing that FUN30 has an annealing activity. So this is the starting template, a single-stranded DNA that is labeled fluorescently. And in the presence of FUN30, it can be annealed to a DNA strand that has sequence homology to part of it. So there is an increase, basically, annealing activity in the presence of FUN30. Through a collaboration with Dr. Samir Atoub's lab at the Department of Pharmacology at UA University, we tried to study the function of FUN30 in mammalian cells and its effect on breast cancer cell lines. So when SMARCAD1, the homolog of FUN30, is knocked down by siRNA, two different siRNAs, we can see that there is a decrease in cellular proliferation, cellular invasion, and mobility and cell invasion as a result of knocking down SMARCAD1. So it seems to have a role in breast cancer. And this is we've used here a breast cancer cell line. In addition to FUN30, we are also working on other chromatin remodelers. One of them is called IRC20. We've purified this using the tandem affinity purification technique. Here it is. And we have been able to show that it can bind to nucleosomes and nucleosomal DNA. So this is a binding assay using an array of nucleosomes and a pull down using magnetic beads, and we can see that basically this protein IRC20 is pulled down to the bead, so meaning that it can bind to nucleosomes. It also seems to have some remodeling activity. This is a restriction enzyme accessibility assay where we can see that with increasing concentrations of IRC20, similar to other chromatin remodelers, you can generate cuts, meaning that the histones are moved out of the way and the restriction enzyme can now access the underlying DNA to digest it. This phenomena is ATP dependent. We also are interested in another chromatin remodeler, a potential chromatin remodeler. It's called IRC5, or LSH5 for lymphoid-specific helicase, or helicase lymphoid-specific. Again, we purify this through tandem affinity purification, and we have shown that it can bind to nucleosomal DNA and nucleosomal arrays as well. We've also shown that IRC5 is required for the activation of checkpoint genes after DNA damage. One of those is RNR genes. So RNR genes are activated with response to MMS, a DNA damaging agent, and the deletion seems to be affecting this expression. So IRC5 seems to be required for the full activation of RNR genes upon damage. Okay, I'd like to acknowledge the people in my lab, Zaina Natur, Dina Muhammad. Those are the two PhD students in the lab, and Jesha, postdoc. I'd like to also acknowledge Dr. Samir Atoub for the work on SMARCAD1 genes, our collaborators, Danish Moazed at Harvard Medical School, Jerry Workman at Stowers Institute for Cancer Research, Tom Owen Hughes at the Wellcome Trust Biocenter, and Philippe Procheson at University of Kansas. I also like to thank the UAU for financial support, Dubai Harvard Foundation, and the Terry Fox Foundation as well. And lastly, I'd like to, for those of you who are students in the audience, like to advertise the graduate program that we have at the UAE University. In different colleges, we have both master's and PhD programs. And specifically at the College of Medicine, we have programs in genetics, cancer biology, molecular biology, and a variety of other programs. So if you have any questions about those and how to apply for those, please let me know or send me an email. Thank you for your attention.
We thank Dr. Ahmed Al-Marzouqi for this interesting lecture. Talking about the epigenetics and how orders can be transmitted out of the genetic code. We'll leave the floor for a question or two according to the time. Yes?
Yeah,
Could you please introduce yourself and then pose the question? Yes.
I'm Doug Higgs from Oxford. I didn't quite understand this. Fun30, you're saying, is a chromatin remodeling factor of the SNF2 family, and it's ATP dependent, but you're actually showing your supercoiling experiment that it's actually not ATP dependent, and it's actually been inhibited by ATP. I don't understand that at all.
Right. The chromatin remodelers, they use the energy of ATP hydrolysis to do certain functions. So, for example, the switch-SNF chromatin remodeler uses ATP to remodel the structure of chromatin completely or transfer the octamer elsewhere. Some chromatin remodelers have additional functions other than that function of ATP hydrolysis that is linked to ATP hydrolysis. So, for example, there are other chromatin remodelers like NO80 that have roles in DNA repair that modify the structure of chromatin using ATP hydrolysis, but also for some other functions do not require ATP. So perhaps the supercoiling assay or annealing assay of Fun30 may not require ATP hydrolysis, whereas chromatin remodeling activity of Fun30 requires ATP hydrolysis. And we have shown that earlier, that was published in that JBC paper. So this is an additional function of this chromatin remodeler, perhaps because of the other subunits that it may have.
Yeah, but the point is, though, that in the DNA supercoiling assay, you must be using energy from somewhere to do the supercoiling. So if it's not using ATP, where's it getting its energy from to do that supercoiling function?
Yeah, so obviously the supercoiling assay, some topoisomerases require ATP hydrolysis like DNA gyrase, and there are other topoisomerases that do not require ATP hydrolysis. So perhaps it's one of those.
Okay, we'll take the second and last question from the middle mic.
Hi, this is Dr. Ghazi Al-Sbeh, King Faisal Hospital, Riyadh, Saudi Arabia. You did mention that the knockout or repression of Fun30 cause cells to become resistant to ionizing radiation. And Could you please elaborate on this and why it become resistant, not sensitive, for example? And what is the implication of that?
It's a good question. Really, I don't know. Perhaps, you know, if it has a role in DNA repair, you know, FUN30 may have a role in DNA repair. So in the knockout or in the knockdown, basically that protein is no longer expressed, and therefore DNA repair may not happen, for example. And so in those cases, for example, the cells would have less basically growth, right, if it has a role in DNA repair. Conversely, if it's affecting the expression of other genes that may have basically other functions, and we're trying to understand what those functions are. It's known that FUN30 is involved in DNA repair. It's recruited to double-stranded breaks, and it's involved in DNA end resections. So perhaps through interactions with some repair machines, or somehow it affects the interactions with repair machines and things like that. Okay. I really don't know why that is, but this is a general phenomena that is observed with UV radiation as well as ionizing radiation, that the cells become resistant when the gene is deleted.
So could it be that the cell is escaping apoptosis or something like that?
Well, in the absence of the gene, the cells grow even more. So the wild type cells die, whereas in the absence of genes. So it seems to have some kind of a anti-repair kind of function. How it works, we don't know. But there are other evidence that shows that the deletion of FUN30 is sensitive to certain DNA damaging agents as well. So
anyway, we thank Dr. Marzouqi.